Today's lecture is going to be about clinical epigenetics. In the clinical space, epigenetics is still relatively new and we are still learning a lot about how it applies to our patients and what sort of tests we need to do, although there are some conditions which are important to know, and there are epigenetic mechanisms that cause some of these conditions. So let's talk a little bit about epigenetics. So basically, epi is a prefix derived from the Greek language. It can mean near, over, among, toward, or upon, like epicanthus, so near the canthus in the eye. In the context of epigenetics, it can be taken to mean in addition to genes, or in addition to genetics. So there are things in addition to the sequence variants that can affect the way a gene functions or manifests itself. So this has something to do with the way genes are expressed or suppressed in different cells or different tissues. Not all genes are expressed in all the cells or in all the tissues. Many genes are not transcribed, which means many genes are not converted into RNA and then subsequently into protein in some of the cells or tissues or at certain stages of development. So they'll have to be silenced at different times and in different organs. For example, some genes expressed in the retinal pigment epithelium are not expressed in the cardiac myocytes. So this makes sense to us. So therefore, there is differential expression of genes based on epigenetics in different tissues and cells, and in different timelines in a person's growth and development. This controlling of expression of genes in various tissues or at the different stages of development is achieved by epigenetic modification. Epigenetics can be defined as any factor that can affect the gene function without any changes to the primary DNA sequence. Some typical epigenetic factors involve alterations in DNA methylation. We'll talk about all of this a bit later on, or the chromatin structure, histone modifications, and transcription factor binding that change the structure of the way the genome sits, a 3D structure, and it can affect the gene expression. Not necessarily the sequence, the structure of the sequence variants is not changed, but the way the genome sits, how it coils itself, how it expresses or expands itself or constricts itself, all of that can be modified in order to make a gene express itself or to inhibit the expression of the gene. Some genes, so there are some other genes other than the genes that get expressed or not expressed. So some genes encode epigenetic regulators. So there might be genes that are located a little bit further away from these methylated genes or the genes that are getting affected. And variants in these genes, so say there is a problem in the sequence variant in these genes, they can actually cause epigenetic disease because they are originally meant to regulate what genes are expressed and not expressed in particular organs or at particular times in development. But when there is a problem in that kind of regulation which comes from another external gene, then there can be an epigenetic disease, and we'll go through some examples later on. So phenotype is the manifestation of a genetic disease. If there is the genome, the sequence variants in a genome, plus the epigenome and the environment, all of these mixed together is what forms the phenotype. And this is what we know at the moment, that it is not just the genome. That's why if somebody's got a sequence variant in a genome, they might either manifest or not manifest a particular condition. But when you add the epigenome and the environmental factors, then you know that there is variation in terms of expression of a particular genetic disease. So we'll be referring back to this diagram. So as you can see later on as well. So let's spend a little bit of time to try and understand what this diagram is saying. So you can look at the chromosome here, and when you expand or when you pull out the DNA from the chromosome, you can see that it's heavily coiled. But if you can pull it out further, you can see over here that there are histones, or the nucleosomes. So around the nucleosomes, we talked in a previous lecture, the DNA gets coiled around it, and there are histone tails present in these nucleosomes, And if there's modification to the histone tail, that can then lead to expression or non-expression of the gene that comes after that histone tail. So this is called histone modification. Another way of changing the expression of a gene is DNA methylation, and we'll look at that in our next slide. But it simply means adding a methylation group or a methylation to the DNA, the CG of the DNA, and that usually represses the gene expression. And there are also various other ways, like histone variants in specific regions, which we will talk about. Before we go into more detail, it's important to understand some epigenetic terminologies. Now, this is not something that's done in a day-to-day. Epigenetics is not something that's used in day-to-day clinical practice, so I'm not going to go into way too much detail, but I'm just going to use things that are clinically important for you. So if you do become a geneticist, or you do become a pediatrician, or you do see some patients with certain conditions in the future, I just want you to be able to know what the relevance is of epigenetics for these patients. But beyond that, we're still learning, and over the years we may know more, but right now this is sufficient for you to understand in terms of epigenetics. So you may hear, if you're talking about an epigenetic condition, you may hear CPG island or CPG site or just CPG, which simply is a cytosine and guanine dinucleotide that can be methylated. So we talked about in the previous slide with the diagram. Methylation simply means adding a methyl group to the cytosine and guanine, and that basically causes DNA methylation and suppresses the gene expression. When DNA methylation occurs in the promoter region of a particular gene, it decreases its transcription. So transcription is how it is expressed. The gene is actually expressed. We talked about it briefly earlier. DMR, or differentially methylated region, a set of CPGs within a defined locus, which differs between samples based on tissue type, phenotype, exposure, etc. So different tissues will have different levels of methylation of CPGs for the same gene. Epimutation is a change in DNA methylation that results in disease, often occurring at an imprinted locus. And we'll talk about a few conditions where epimutation can be important. Now, what is imprinting? It's really important to understand imprinting because there are some medical disorders of imprinting. Imprinting is a process whereby only one copy of the gene is expressed, either maternal or the paternal copy, and the other copy is silenced by methylation. So only one copy of the gene is expressed, and if that is the case, it is called imprinting. Normally both copies are expressed, but when there is imprinting, only one of those copies is expressed. Epigenetic modification occurs throughout life, starting from embryonic and fetal development. For example, cells divide and differentiate into distinct tissues and organ-specific cell lines in the embryo. That's because of epigenetic modification. Epigenetics is especially important in early embryogenesis. So this epigenetic modification decides the rate of growth. There's differential rates of growth, or organ-specific epigenetic modification occurs. So different types of organs are formed, and not all genes are expressed in all organs, for instance. Environmental interaction with the epigenome during in utero stage can also have effects on later health outcomes because of some epigenetic sort of memory that does occur in that particular gene. For instance, in assisted reproductive technologies like IVF, there can be an increase in some imprinting diseases. And prenatal starvation can increase later life incidence of obesity, cardiovascular disease when the child becomes an adult. So these are all due to epigenetic modification caused due to environmental interaction. We know a lot about epigenetics through twin studies. DNA methylation patterns across the lifespan is best observed in monozygotic twins. So as you know, monozygotic twins share the DNA, so they have the same DNA, and they're born with not identical, still non-identical, but highly concordant DNA methylation patterns, which means that their DNA methylation patterns are very similar. But following birth, particularly if they're raised in slightly different environments, then the DNA methylation patterns of these monozygotic twins become increasingly divergent with age. So this likely occurs in response to ongoing environmental differences and the differences in methylation patterns in cells due to this. So what is a genomic imprinting disorder? In some genes, the imprinted genes, only one copy of the gene is expressed, either the maternal copy or the paternal copy. Normally both the copies are expressed, but in the imprinted genes, only one of these copies is expressed. Imprinting disorders occur due to disruption of the normal monoallelic expression of these genes. In other words, instead of just one copy being expressed, both the copies may be expressed, which will cause a disorder, or neither copy is being expressed, which will also cause a disorder. The majority of imprinted genes are found in clusters called imprinted domains in specific chromosome regions, and we can talk about some examples later on. Each imprinted domain is controlled by one or more independent imprinting control region that regulate in cis, which means on the same side of the chromosome. It doesn't control the opposite chromosome. So that regulate in cis the expression of target imprinted genes within the domain. Epigenetic changes can impact imprinting centers. This is called epimutation. Now, the last point is important, so please pay attention. Clinically, if you want to find out if your patient's got an imprinting disorder, if you organize any fancy test like a targeted panel sequencing test, whole exome test, or even a whole genome test, you won't find the answer, because this is not, generally speaking, a sequencing disorder. So it's not a problem with the variant within the gene itself, although in some cases you can have a variant detected in certain genes that control these imprinting centers. In most cases, what you want to find is if the gene actually has a methylation problem. So is it actually expressed or is it not actually expressed? Are both copies expressed or are both copies suppressed? Therefore, a special type of test needs to be organized, and the test that is usually clinically ordered is called a methylation sensitive multiplex ligation dependent probe amplification, otherwise also called MSMLPA. So it's really important to know that you can't simply do a sequencing test like a targeted sequencing panel or whole exome or even whole genome sequencing to detect a genomic imprinting disorder. It has to be a methylation sensitive type of test. Some examples of genomic imprinting disorders include Prader-Willi syndrome and Angelman syndrome. So these were the first human disorders recognized to result from genomic imprinting. We will discuss these in case discussions at the end of our lecture series, but I'll introduce you to some very basic details of this, basic snippets of this condition in this slide. So these two neurodevelopmental disorders result from the absence or mutation of paternally or maternally inherited copy in the 15q11 to 13 imprinted region, which contains a number of imprinted genes. So imprinted genes, reminding you again, is only one of the copies is expressed and the other copy is not expressed, either the maternal copy or the paternal copy is expressed and the other copy is not expressed. But say there is a case where either the maternal copy is affected or the paternal copy is affected, therefore it's not doing what it's meant to be doing, then you can have an imprinting disorder. In the case of Angelman syndrome, the maternal copy is affected. And in the case of Prader-Willi syndrome, the paternal copy, or the one that comes from the father, is affected. So why is it affected? What happens? Why is either the maternal copy or the paternal copy affected? Why doesn't it do what it's normally meant to do? So this can occur due to many different mechanisms. So there are different ways that either the paternal or the maternal copy can be affected. There could be chromosome deletion. So this part of the chromosome, either containing the maternal copy or the paternal copy, can be deleted because of a particular genetic disorder. Or there can be uniparental disomy. So chromosome deletions usually occur, I think you may have learnt in some of your previous lectures about recombination disorders, and that's where chromosome deletions can occur due to unbalanced recombination events. Uniparental disomy is where two copies of a chromosome from the same parent are inherited. So that's a meiotic disorder. If you go back to the lecture about meiosis, instead of inheriting one copy of chromosome from the mother and another copy from the father, sometimes there is a disorder where both copies are inherited from the mother or both copies are inherited from the father, which just means either the maternal copy is not present or the paternal copy is not present, depending on which two parental chromosomes have been inherited. And that can cause a genomic imprinting disorder. There are also epimutations, so mutations that occur in the imprinting domains, and pathogenic sequence variants in specific genes. So for instance, in Angelman syndrome, there is a gene called UBE3A, so we've identified that sequence variants also in this gene can cause Angelman syndrome. So this is not an this causes decrease in the expression of that particular area of the gene, of the cluster. There's increased risk in babies born through artificial reproductive technologies of genomic imprinting disorders, as we talked about earlier. Another example, and I thought this is a nice diagram from your textbook, is Russell Silver and Beckwith-Wiedemann syndrome. So if you look at the middle section with the rectangle around it, that's normal imprinting. Okay. In this case, in this particular area, so you can see the paternal area of the imprinting center 1 is methylated, which means it's silenced, and the maternal area is actually not silenced, so it's unmethylated. So the maternal is expressed and the paternal is suppressed, and that is normal, and you have a healthy baby. But if you look above that rectangle, you can see here Russell-Silver syndrome, where both the paternal and the maternal copies are unmethylated, which means both are being expressed. And this causes an undergrowth type syndrome. The baby is very small and it has growth deficiency and other problems as well. And that causes Russell-Silver syndrome. And it can only be detected by doing special type of testing to see if there's methylation problems. On the other hand, the opposite end of the spectrum is Beckwith-Wiedemann syndrome, where both the paternal and the maternal copies are methylated, which means they're both silenced, they're not expressed, and there can be an overgrowth syndrome called Beckwith-Wiedemann syndrome, which also has a lot of other issues as well. So as you can see, this is what happens in imprinting disorders based on how the genes are expressed, differentially expressed. There are also some other epigenetic disorders, but I'm not going to go into too many details about these, as these are not considered so clinically relevant at this stage. However, disorders involving unstable repeat expansions like Fragile X syndrome can also cause epigenetic problems. So full mutation in Fragile X syndrome results in methylation of the FMR1 promoter, and therefore there's no transcription of the gene. Many cancers can also result in epigenetic problems, and a number of neurodevelopmental disorders are due to epigenetic problems as well. With this, I'm going to stop this topic of epigenetics here. It can be quite a lot sometimes and may not be necessarily completely relevant, but there are some important aspects of epigenetics which are clinically relevant. So what I've discussed so far is relevant to you, and please go through these slides again, and if you've got any questions, as usual, please email education@pansma.org, and we will try and send you back answers to your questions. Thank you.